you're happy for that says that you're happy for your little initials to be recorded and shared afterwards. There we go. So welcome everyone to the latest lecture in the OSME series. I will, without further ado, hand you over to Lee for today's lecture. Enjoy.
Thanks very much, Harry, for the intro. And yeah, very good afternoon, everybody. First things first, can you all hear me okay? If you just pop a quick yes in the chat, or if you feel free to feel confident to you can. Lovely, thank you very much. Okay, so I hope you're all doing well, guys. So firstly, just to introduce myself, as Harry said, my name's Lee, and I'm one of the fifth year medical students, and it's my great pleasure to be here delivering this lecture as part of the Oster Society of Medical Education's National Preclinical Revision course. So for those of you that have attended the lectures so far, I hope that you've enjoyed them, and I hope that they've been useful for you and just sort of your revision going forwards. So my hope today is to deliver what will be the first of sort of two biochemistry lectures. So the first will be obviously today on proteins, lipids and carbohydrates, and the second one will be next Saturday on glucose metabolism. And I guess sort of what I hope to sort of focus on is primarily areas that are, you know, quite likely to appear in your preclinical exams. So, you know, focusing mainly on sort of high yield concepts because, you know, as medical students, we all appreciate a bit of high yield information. So that will be sort of the main focus. And we can also obviously go through any questions that you have along the way if you feel confident enough to unmute, or if not, then just pop a question in the chat and we can revisit that at the end. That's not a problem. So really quickly, just sort of housekeeping. I'll be aiming to deliver this hopefully within the hour. And I know sort of many of you have given up your sort of hard-earned Saturdays to attend, so I really want to be respectful of your time and stick to that. But sort of do forgive me if we do overrun, but if we do, it shouldn't be by more than sort of 5 to 10 minutes anyway. I think that's everything. So with that said, and without further ado, let's get started. Okay, so first up on our sort of tour through the molecules of life, as it were, we're going to start with proteins, which I hope that you're all familiar with from your sort of pre-medical school studies, shall we say. But hopefully I can also introduce some sort of new concepts that you might not be familiar with too. So we'll start with the building blocks of protein structure, so amino acids and amino acid chemistry. So hopefully the sort of the general structure of amino acid is familiar to you. So essentially, as you can see on the slide here, what we've got is a sort of a central alpha carbon atom here attached to a variety of sort of different groups. So we've got a carboxylic acid group on the right there, which is the acid component of the amino acid. We've got an amine group here, which obviously is the amino group. So together they make up the amino acid. We have a single hydrogen atom here that's also attached to the carbon. And then importantly, we have this sort of R group here, which can be a variety of different chemical structures and is what differentiates the individual amino acids according to their different physiochemical properties within the cell. So essentially, this amino acid here has been drawn with the carboxyl and the amino group in their sort of neutral states. But in terms of at the physiological level, so if we're thinking about physiological pH, about 7.4, these groups will very likely be either protonated in the form of the amine group here, or in the deprotonated form in terms of the carboxylic acid group. And that can obviously influence, you know, the amino acid in terms of its behavior, but also the environment that the amino acid finds itself in as well. So in terms of genetically encoded amino acids, there are 20. as far as we know, and you can see these just on the slide here. And obviously these amino acids are genetically encoded. That is that the amino acids are encoded by the mRNA transcript and assembled by the ribosome to form a growing polypeptide chain. Now, I wouldn't advise you to go away and learn these structures by rote. Of course, if you're interested in doing so, that's absolutely fine. But I think what's more important here for you all is just to appreciate the sort of the physicochemical properties of the amino acids based on their different structures, and to be able to recognize the different types of amino acids if you were to see one. So, for instance, we have the polar amino acids that will have polar groups, so charge groups that are able to form ionic interactions, and we have the hydrophobic amino acids, which are predominantly sort of carbon-carbon-carbon hydrogen bonds in terms of their chemistry. And essentially, all of these different R groups that the amino acids have will influence the chemistry and the behavior of the amino acids, and essentially affect the function of that, not just that amino acid, but the protein that the amino acids find themselves in. I guess I should also mention that all the amino acids have 3-letter codes, which you can see here, which sort of obviously denotes which amino acid it is, as well as 1-letter codes as well, as a shorthand, which is often quite useful when they're reported in terms of a very long peptide sequence. So in terms of the amino acids themselves, hopefully you're all sort of familiar with how they can be assembled, and we'll talk about that in the next few slides. But just a quick recap. So the sort of linear assembly of amino acids into a sort of defined sequence is termed the primary sequence of the protein, and that is principally held together by covalent or peptide bonds, and that can be sequences of sort of various lengths. The primary sequence will then determine the overall fold of the protein, and then the protein fold will determine the function of the overall protein as well, in terms of how it operates at the biological level. So when we think about growing the polypeptide chain, so we're thinking about linking amino acids together. Again, I hope this is familiar to you, but essentially when we form a peptide, we have 2 amino acids together, and essentially we have a reaction here called a condensation reaction, where the carboxylic acid group of one amino acid and the amino group of the other amino acid undergoes a reaction known as condensation, where you have the elimination of a water molecule and the formation of a peptide bond, a covalent link between those 2 amino acids to form the dipeptide from the 2 original amino acid monomers. Now, by convention, it's important, I think, to note that peptides are generally written N to C terminus. So the N terminus being the amino acid, the amino group of the amino acid at the start of the protein, and then the C terminus being the carboxylic acid group of the end amino acid of that protein. And that's just a really helpful convention to sort of follow when we're managing or dealing with long peptide sequences, and we want to be able to identify which amino acids are at the start and which amino acids at the end. I guess also on this particular slide, a slightly minor and subtle point, but I think it's still probably worth mentioning. So, as I said, when we condense these 2 amino acids together to form the dipeptide, which is linked by a peptide bond, it's important to appreciate from a sort of a structural perspective that many of the bonds within this dipeptide here are going to be fully rotational, or at least have a large degree of flexibility around them. And that is true for most of the bonds in this dipeptide, with the exception of the peptide bond, which has quite a unique property in that we call it a partial double bond character. So I won't go into the details, but if you're interested, of course do look it up. But essentially what we mean by this is that if you look at this sort of amide peptide bond here, the lone pair of electrons, which will be on this nitrogen, can be donated to this carbon-nitrogen bond here, and essentially can make the carbon-nitrogen bond a partial double bond. So the electrons can move around this group here, and essentially that restricts the rotation of that peptide bond, so that it's not able to rotate as freely. And obviously this has real implications for protein structure going forwards, in that if peptide bonds are inflexible, then essentially that can help to define how the primary sequence is going to be oriented and the geometry of that relative to the rest of the protein. An analogy I quite like here is thinking about spaghetti. So, if we've ever, if any of you have ever cooked spaghetti, you might find that when it's not quite cooked, there are some areas of the spaghetti that are a little bit softer, and there are other little bits within the spaghetti strand that are perhaps a little bit harder, a bit more chalky. And I kind of think that that's quite similar to what we're explaining here, in that there's the sort of the chalky bits that are a bit more rigid. We can think about those as the peptide bonds, and that's going to affect how our protein is going to sort of be structured in solution. So we've talked about amino acids. We've talked about how they react together to form a dipeptide. Now we're thinking about growing that peptide chain into a polypeptide, and so you can see on the left here that many amino acids assembled into a linear sequence can form the growing polypeptide chain. And this is essentially a space filling model, which some of you may have seen before. So this is just a sort of a structural representation of the what the atoms would potentially look like in this particular protein or polypeptide. And essentially, when that polypeptide that you can see on your screen there undergoes the process of protein folding, we form this globular structural unit, which is a protein with a defined 3D structure and a defined function. And I guess just as a side note, this is something that's been quite topical of late. Over the past 50 years, it's been very it's been an area of real growth in terms of how can we go from predicting the protein's 3D structure and function from the primary sequence alone. And essentially that's been a question that computational biologists have found to be relatively straightforward for smaller proteins, but for bigger proteins, where it becomes a bit more complicated to predict protein structure from the initial sequence, some sort of more complex computer programs might be more helpful. And you may have seen in the news about DeepMind, who is an AI company related to Google, have developed systems, machine learning software that can quite accurately predict what the 3D structure of a protein is going to be like from this initial structure. And that's really, really important for anyone really working in medicine, because it saves us a lot of time, particularly when we're developing drugs, because as I'm sure you're aware, most drugs are targeting some sort of cellular process, and ultimately those targets tend to be enzymes or proteins. So there's a lot of time to be saved there if we can predict what the protein structure is going to be like without actually having to express the protein and to determine the structure by other biochemical methods. And again, so we're thinking about protein structure, or the folds, and we can think about this, as I've alluded to already, at four sort of different levels. So we've got the primary sequence, which is based on the covalent linkage of amino acids. We've got the secondary structure, the tertiary structure, and the quaternary structure, all of which I'll go into more detail on in a moment. But essentially, these are all sort of higher order structures of the protein that are formed from further folding of the primary structure. So the formation of secondary, quaternary, and tertiary structures of proteins are from the primary structure are almost all driven by non-covalent interactions. And these interactions are really, really important, particularly because they allow biological systems to have dynamics, and it's so important that they are able to that these forces are able to be made and then readily broken and then remade in order to help the reactions that they're catalyzing. And they're also essential. These forces are essential to sort of DNA, lipid bilayers, proteins, and obviously there are many different types of forces here. So I'm not going to labour the point because I'm sure that you're all well aware of this. But essentially, we've got ionic interactions here, which are attractions of opposite charges. So oppositely charged amino acid side chains. We've got the hydrogen bonds here, which are the proton donors and acceptors between hydrogens on electronegative atoms with other electron pairs. We've got hydrophobic interactions with polar groups, with non-polar groups that are water repelling or water hating. And then also we've got van der Waals forces, which play more or less a less significant role here, but are interactions between weakly polarizing bonds. So just go through secondary structure very quickly. So this is driven by, as I said, hydrogen bonding between the backbone NH groups of peptide bonds. And essentially the secondary structures can be sort of broadly split into two different camps. So we've got the alpha helices and the beta sheets here. So in terms of the alpha helices, we have peptide bonds that form in a manner that's sort of perpendicular to the axis of the helix here. So we have the peptide bonds that form linearly as the helix goes down, and that's primarily or exclusively between the backbone NH of peptide bonds. The other is the beta pleated sheets, which again I'm sure you may have heard of, and these can exist in two forms. You've got the parallel or the anti-parallel, and essentially the beta sheet is an anti-parallel if the strands run in opposite directions, which you can see here on the diagram, going from C to N and then N to C. And then you can also have parallel strands, and this is just strands that run in the same direction that will give a slightly different structure. Also on the diagram here, you might be able to appreciate, it's not labeled, but there's also secondary structures within the actual protein structure itself. So if you look at the two beta sheets on the diagram, they're connected via a little loop region, which is known as a beta turn. And these are really important for connecting anti-parallel beta sheets and facilitating the hydrogen bonding interactions that occur between those beta sheets. Again, just to sort of highlight the point about tertiary structure, really, really important to be able to sort of visualize and pick apart these in the actual structure themselves. Hydrophobic interactions that can form within the polypeptide core because they want to avoid contact with water. We've got the disulfide bonds, which are formed through the side chains of our cysteine amino acids. And I guess the important point about the disulfides is that rather than playing a role in driving the folding of the protein, a way I like to think of this is that they act as almost like molecular staples. So the tertiary structure of the protein is driven primarily by the hydrogen bonds, the hydrophilic interactions, and the hydrophobic interactions. But then your disulfide bonds are almost acting as the staples which hold everything together and stabilize that structure so that it's nice and secure. And finally, we've got sort of the the top of the protein structure hierarchy, which is the quaternary structure. And this is essentially a structure that's made up of multiple discrete, independently folded protein polypeptide chains, which are each held together by non-covalent interactions. And the prototypical example of this, which I'm sure you're all aware of, is hemoglobin, which you can see on the slide here. So hemoglobin is a alpha-2 beta-2 tetramer, so it's made up of four subunits, 2 alpha and 2 beta, and these are each independently folded polypeptide chains, and we'll explore in a bit more detail later on about the importance of this structure in terms of understanding the function of hemoglobin as a molecule. So protein functions. Proteins are the workhorses of the cell, so they have a variety of diverse roles in cells, and we can sort of conceptualize the functions of proteins into different categories. Firstly, we can sort of think about structural proteins, and specifically for this talk, I'm going to focus on collagen in the next few slides. We've got transporters, so these are obviously proteins very much like hemoglobin and oxygen transporters like hemoglobin and myoglobin, but also transporters that are integral membrane proteins that are embedded within the cell membrane, which facilitate the transfer of different cargo into and out of the cell. And often this is charged species, metabolites that would otherwise normally not be able to passively diffuse through the cell. And then obviously we've got enzymes, which are our biological catalysts that facilitates the majority of the chemical reactions within the cell. Now, just to take you through the different protein structures here at the bottom of the slide. And so these are sort of structures, so experimentally determined protein structures. So on the left here, you can see we have what's called a transcription factor that binds to specific DNA target sequences called promoters. And essentially the role of that transcription factor is to form a complex with other proteins that enable essentially gene transcription to produce mRNA transcripts and eventually protein products that can be used by the cell to facilitate the functions of that cell. The picture on the center here is again a membrane receptor which is embedded within the membrane bilayer. And these are often critical for relaying signals from the outside of the cell to the inside of the cell, often in response to a change in the cellular environment and enabling the cell to respond in a particular way. And finally, the one on the right here is a protein which you may have heard of, and it's called ATP synthase. And I unashamedly had to include this as it's my favorite protein for one. Yes, I do have a sad life. But seriously, if you do have a few minutes, I encourage you all to look up ATP synthase. It has a fascinating mechanism of action. And essentially this is that the protein works by coupling the electrochemical gradient, the proton gradient in your mitochondria that's developed through the process of oxidative phosphorylation, and this is coupled to the synthesis of ATP. And essentially this protein does this by acting like a little molecular turbine, so it spins in the cell, which is that rotary part at the top there. And I just think the idea of millions and millions of these happening in your individual mitochondria in your cells that are spinning and producing ATP every second of your existence is something quite magical, really. So I think it's a very, very interesting and cool protein. So just to start with a few examples of some structural proteins, and the main one that I'll discuss and that's in the syllabus of the preclinical course is collagen. So essentially collagen is a structural protein, and it's the most abundant protein in the human body. Structurally, essentially collagen has evolved primarily to provide mechanical stability to various tissues, joints, bones, and tendons. And there's multiple different types of collagen that we can find in different tissues, which in essence are sort of fundamentally quite similar, but can be subtly different in that each is adapted for function in that specific tissue. Now, the important point about sort of collagen and structural proteins is that any changes in protein structure can lead to human disease, essentially. And in collagen, this is a particularly notable example. And there's a couple of sort of disease pathologies collectively known as the collagenopathies that really highlight this point quite well. And so essentially, if you have defects in the structure of collagen, particularly through single amino acid changes in the primary sequence of the protein, then this can either act to destabilize or stabilize the tertiary structures to have a major effect on that protein function. And what we can see here on the right is an X-ray of a child with a condition called osteogenesis imperfecta, less formally known as brittle bone disease. And essentially what you can see there in that X-ray is you can see that the long bones in the upper arms, the humeruses, are essentially all quite irregular because the bones are quite brittle and can actually break in utero. So a lot of babies with osteogenesis imperfecta cannot be born through the birth canal because it would just simply crush the bones because they're so fragile and just so delicate. So this really is a condition which has a huge impact on the quality of life of patients that are unfortunate enough to suffer daily with these problems. So just going into a bit more detail about, because I know that collagen is specifically on your syllabus. So the exact defect in collagen is a defect in the helix itself. So collagen, in terms of its primary structure, consists of a helix that's a regular helix, and that helix is made up of a glycine, proline, and any amino acid repeats. So it's a glycine proline repeat sequence, and that then goes on to form a single helix. Now, what gives collagen its real strength is that these single helices can associate into essentially trimers, so bundles of 3 helices, to form triple helixes to maximize their tensile strength. And essentially, it's this helical structure that can then be wound together into higher order bundles or higher order macromolecular structures to form individual collagen fibers. And you can see this pretty clearly on this diagram here in the electron microscopy image, which shows a really beautifully ordered structure to collagen, which enables it to have this function of high tensile strength. Now, specifically with collagen, there are many defects that you can actually find in the protein, but I guess the prototypical one that I think it's important that you guys are aware of is the glycine to alanine mutation. So essentially, in the collagen helix, it's very often the case that you find glycine residues in the at the corners of the helix, so just as the helix is about to turn. And I guess you can think this is sort of quite logical, given that glycine has probably the smallest side chain of all the amino acids, so it has just a single hydrogen. So it's very easy for glycines to allow that sort of tight turning of the helix and contribute to that tensile strength. However, if you have a mutation where you substitute the glycine to an alanine, which has a CH3, a methyl side chain in the R group, that then adds a little bit more bulk to the side chain, which means that it's harder for the helices to form these lovely tight turns that enable the protein to have this high tensile strength. And so essentially what you end up having is a helix. As a result of this mutation, you have a helix that doesn't pack together as tightly, and a protein that has reduced tensile strength and a mechanical strength that's ultimately been compromised, which is linking it back to our patients. is why they have bones that are extremely fragile when a minimum amount of force is applied to them. So another example of protein structures or alterations in protein structures that can lead to disease is hemoglobin. And again, I think the classical example of a sort of mutation in hemoglobin that's of sort of pathological relevance is sickle cell disease. So essentially, hemoglobin, the dominant protein in red blood cells, and during red blood cell maturation, the nucleus is lost during the sort of the terminal differentiation stages of the red blood cell to essentially allow for the red blood cell to pack itself with more hemoglobin to maximize its oxygen transfer capacity. Now, in diseases such as sickle cell, essentially the key mutation to be aware of is a glutamate at position 6 to a valine, and these mutations are happening exclusively in the beta globin subunits. So these are the beta, not the alpha subunits. And essentially what you're doing here is that you're producing a red blood cell that's structurally unstable. You're going from a highly polar residue in the glutamate to a valine residue which is very, very hydrophobic. And so essentially what that produces as a result of that mutation is a sickling of the red blood cells, and a distortion of that classical sort of discoid shape that we all know and appreciate of a red blood cell. Now, once the red blood cells acquire this mutation, the hemoglobin molecules associate into clusters, and hemoglobin then starts behaving not as this sort of organized quaternary structure, but as long inflexible chains, and essentially become sickled in shape as a result. And this is a real problem because red blood cells have evolved obviously to travel through capillaries to maximize oxygen diffusion to tissues, and sickled red blood cells become clogged in capillaries, and can produce really painful joints in fingers of patients that have sickle cell disease. So obviously this is another condition which is important to be aware of, but also important complications that patients that can have, and I'm sure that this is something that you'll become more aware of as you progress throughout your medical school careers and go into clinical school. So that's the first part of proteins done. So we've covered protein structures and covered protein function with an emphasis on collagen and hemoglobin. The next thing I'd just very briefly like to touch on is enzyme regulation and catalysis. So I'm not going to labour the point too much here, but essentially enzymes, biological catalysts that increase rates of chemical reactions. And essentially we can sort of formalize their function as happening in this kind of defined reaction pathway here. So we have an enzyme that can bind a substrate of specific structure that's complementary to the active site. Some chemistry goes on there between the enzyme and the substrate to facilitate a reaction which then produces a product, but then ultimately and importantly enables the enzyme as a catalyst to be regenerated from the reaction. And super important that this happens because if we didn't have enzymes, then just these very simple chemical reactions like making and breaking covalent bonds could take years to complete, rather than the microseconds and the nanoseconds that we need them to be on the molecular level scale. So again, this is just hopefully some revision about enzyme catalysis. So the way that enzymes are working in the cell to increase rates of chemical reactions is to stabilize the transition state by lowering the activation energy and allowing the reaction pathway to take an alternative reaction route to enable the reaction to proceed more quickly and accelerate its progression. And I guess enzymes, one question you may be asking is, well, how do they do this? What mechanisms do they employ to lower the activation energy of the reaction that they're catalyzing? So you can either have enzymes which orient the substrate in a certain way to optimize the chemistry of the reaction. So there's lots of dynamics that are happening there with in the active site of the enzyme. There may be some physical strain that the enzyme imparts on the substrate, so it may be able to distort some of the bonds that need to be broken, which render them more amenable to being reactive. And I guess another really important consideration is that enzymes are able to stabilize the formation of intermediates in that reaction between the substrate and the active site. And stabilizing those charged intermediates is going to really help your reaction to proceed much more efficiently as well. So when we think about enzymes and measuring their activity, one of the most well-known models that I think is important to be aware of is so-called Michaelis-Menten kinetics, named after its co-founders, Michaelis and Menten. And essentially, the concept here is that this model is describing the rate of an enzyme reaction relative to the concentration of substrates. And importantly, what we get from Michaelis-Menten kinetics, what it tells us is that we have two very important parameters of enzyme activity, and that is the Vmax and the Km. So as you can see on the graph here, so the Vmax is essentially the maximum rate of the enzyme catalyzed reaction. It's when the enzyme's working at the maximum turnover rate. The Km is known as the Michaelis constant, and that is equal to half the substrate concentration. So essentially that's when the enzyme is turning over substrate optimally, but not at its maximum rate, essentially. Okay, and as we can see sort of from this curve here, if we plot reaction rates over substrate concentration, at the higher substrate concentrations, the reaction rate kind of tapers off and becomes limited, and that's due to the number of available active sites becoming limited, which is why we see the curve eventually flattening off there. So enzymes, as we know, can be influenced by inhibitors, and inhibitors come in a variety of different flavors, essentially. And I guess a good way of thinking about that is to split them up into competitive and non-competitive. So in terms of competitive inhibitors, we can think about these as inhibitors that compete with the substrate for the active site to bind the active site. And essentially the important fact here is that interaction between inhibitor and enzyme active site is actually reversible, and that if we wanted to restore that reaction and to continue the turnover of the enzyme, then we would need to add in more substrate to outcompete the inhibitor, or just remove the inhibitor entirely. So that's competitive reversible inhibition. If we think about its counterpart, so the competitive irreversible inhibitors, this is, as the name suggests, where you have binding of the inhibitor which is irreversible, and essentially in most cases this is a covalently linked inhibitor to the active site. So essentially you're sort of permanently changing the chemistry of that active site, such that the only way that you're able to allow that reaction to progress any further is adding new unaltered enzyme, because you've essentially permanently changed that structure. Another important type of inhibition is non-competitive inhibition, and this is sometimes sort of thrown into the group of allosteric inhibition as well. So this is inhibitors that are binding at alternative locations on enzymes away from the active site, and indirectly the binding of these inhibitors are altering the structure of the active site and perturbing substrate binding. So just to follow on from allosteric inhibition and regulation, and introducing this concept of cooperativity. So essentially, cooperativity is a phenomenon that takes place particularly within multimeric proteins, such as hemoglobin, and is essentially referring to the impact of one subunit of the protein upon binding either a substrate, a ligand, or an effector on the behavior of other subunits. And so we can think of cooperative binding as really as a special case of allosterism. Okay, and essentially if we use sort of hemoglobin as an example, hemoglobin is a perfect example of what we call positive cooperativity. So essentially, this is a process that occurs as a result of binding oxygen to produce a successive increase in the affinity of oxygen binding at other heme groups. So essentially, in cooperative binding of oxygen to hemoglobin, we get binding of one oxygen to one subunit. That inadvertently increases the affinity for subsequent oxygen binding at other sites, until eventually you reach a hemoglobin that's fully saturated with oxygen. And this is super important for hemoglobin function, because essentially what we want from hemoglobin is essentially two things. We want to ensure that it can perform to facilitate maximum oxygen uptake from the lungs where it's needed, where oxygen availability is typically particularly high. And also we want hemoglobin to be able to release oxygen to peripheral tissues when oxygen levels are quite low. And it's this quaternary structure and this cooperativity mechanism that enables hemoglobin to perform these particular functions pretty well. So just moving on to thinking about how enzymes might be regulated in other ways other than inhibition. So in this table here, I've just summarized sort of four main ways that this might happen. And essentially, the key ones here are listed. So one way is, which I'm sure you're aware of, is sort of covalent modification to the enzyme, which is often reversible. And classically, the main example that is important to be aware of is phosphorylation. So this is reversible attachment of phosphate groups onto proteins or enzymes to either inhibit them, so to sort of slow down the activity of the enzymes, or it can have the opposite effect of actually upregulating the activity of the enzymes, depending on which system you're looking at. And this sort of mechanism of phosphorylation is sort of widely used across higher organisms, and is most commonly actually seen in signaling pathways involved in cell growth, involved in cell death, and differentiation as well. Proteolytic cleavage is another really important one, and one that I'm going to touch on with an example in a moment. But essentially, this is where you have activation of inactive precursor enzymes that become active after proteolytic digestion. And this is really commonly seen in a variety of biological systems, the most common one being digestive enzyme regulations in our stomach. So when we think about proteases in our stomach, so pepsin and trypsin, both of these are secreted as inactive precursor molecules, pepsinogen and trypsinogen, and secreting them in the active form would not be a great idea, because that would then lead to self-digestion and autolysis. So we really need these proteins to be produced in the inactive form, so that when we need them to perform their roles, they can then be activated appropriately and perform the role that they need to. Allosteric regulation will be probably something that will be more relevant next week when I talk about glucose as a metabolic fuel. But essentially, this is, as we've discussed, small molecules that are binding to alternative sites and enzymes to either repress or upregulate enzyme activity. And finally, we've got isozyme regulation, which is essentially isozymes are enzymes that sort of catalyze the same reactions but will differ very slightly in structures. And that's due to the way that the enzymes are produced during transcription. The way that they're spliced by the spliceosome produces a protein that is different, but is essentially a variant of the same enzyme. But allows that particular enzyme to perform that particular role, which is the same reaction but just in a different tissue. So just to give an example of the importance of one mechanism of enzyme regulation, and that is proteolytic cleavage here. Okay, and essentially one of the key examples of this in action is blood coagulation. Okay. And essentially what we see in blood clotting or coagulation, we have a really tightly regulated process that is really only primed to become active when we have a stimulus. So for instance, in terms of blood clotting, that might be a cut to our finger or our arm, any sort of injury to the endothelial tissue which sort of lines our vessels. That's when this process should be active. And so if we think about, you know, our coagulation system, you know, it wouldn't be terribly great for us humans to have a system which just clots in an unregulated manner when it wants to. That would be pretty unfavorable and would leave us quite vulnerable to blood clots in the lungs and the heart, which you know, is not going to be a good thing. And so, you know, we need to evolve a coagulation system here to help this process become regulated. And one of the ways you can do this is through enzyme activation through what's called zymogens. So zymogens is kind of the full name, so to speak, of an inactive protein precursor that's activated by specific proteolytic cleavage. And when we activate these zymogens, we form essentially an amplification reaction where multiple different types of these activated enzymes can then go on to activate further downstream proteins, and we get amplification of this signal in a very quick manner, which is really, really important because that's exactly what we need when we have a breach to our blood vessels and to form essentially a clot or a hemostatic plug, which results in the early repair of that particular blood vessel. So that's proteins done. I hope that was clear. The next thing we'll move on to then is lipids and lipids structure and function. So what makes something a lipid? So lipids typically very, very chemically diverse structures, particularly rich in carbon-carbon and carbon-hydrogen bonds, and that then gives them their classically sort of hydrophobic nature. So they obviously have quite a tough time dissolving in water. And that's obviously very evident if you've ever added oil to water, and you can see that the two form a partition. They partition into different layers because oil, due to its hydrophobicity, you know, doesn't like associating with water, which is a highly polar solvent. And essentially what we can see here on this slide is sort of the different forms or structural forms chemically that lipids can take. So we have so cholesterol here, which is the precursor for a majority of steroids, which I'll talk about in a moment. And we have triglycerides here, which is you know, obviously a key storage form of lipids. We have a phospholipid structure here, which is obviously an essential component of not just biological membranes, but other parts of the cell. And then we also have free fatty acids molecules here as energy sources and storage molecules. So if we move on to looking at fatty acids can be defined as being what's called amphipathic. So essentially all that means is that they've got the best of both worlds. So they've got this hydrophobic region here, which tends to avoid water and polar solvents, or you've got the hydrophilic group here, which is the carboxylic acid group, and essentially both of those together mean that these species are amphipathic. And when we're thinking about fatty acids, our chemistry also comes in really, really helpful here. So we've got our trans fatty acids here, which is referring to our carbon-carbon double bond, and the groups being attached at opposite ends. And you may have heard of trans fatty acids being particularly relevant to coronary artery disease and increased production of low density lipoproteins sticking to the walls of your coronary vessels and contributing to the coronary artery disease, which is why some dietary guidelines ask or advise to try and limit the consumption of trans fats where possible. And we've also got our cis fats here, which are the fats that are particularly rich in nut oils And as similar to the trans, we have a cis configuration here around the carbon-carbon double bond, which is really, really important for producing this kinking of the structure. So it's going to affect the chemical properties of this particular lipid. So phospholipids, again, are also amphipathic molecules, so consist of, as I'm sure you're all aware, a hydrophobic and hydrophilic moieties. So the precursor for phospholipid synthesis is the glycerol molecule here, and essentially the formation of the phospholipid occurs through esterification of this glycerol molecule, particularly through these 2OH groups, which are substituted with 2 fatty acids, and then that leaves the remaining hydroxyl group of the glycerol molecule to react with a phosphate group that's linked to a different chemical moiety. And we can see that here with this phosphatidylcholine, which is one of a handful of different phospholipids. We have the 2 fatty acids here that have been substituted, and we have a phosphate group that's linked to a particular moiety here, this amine group up the top. And I guess what's really important about phospholipids is that they spontaneously, in aqueous solution, can spontaneously assemble into these supramolecular structures, these so-called lipid bilayers that surround cells. And these can take one of 3 forms. So they can take formation of liposomes. They can be micelles, which are essentially a smaller version of liposomes, or they can be a membrane phospholipid bilayer, which we know is essential for defining the boundaries of cells. And essentially, whether a phospholipid forms each of these structures is largely due to the chemical nature of the fatty acid side chains themselves. So really, really important to consider the side chains of the fatty acids when you're thinking about what structures that they're going to perform. So as I alluded to just a moment ago, so phospholipid classes. So these are the different phospholipid classes that we know of at the moment. And essentially, we have on the left here, we have our big phospholipid molecule that has been substituted by 2 fatty acids, and then the remaining hydroxyl has been phosphorylated, and then attached to that phosphate we have an X, which is a functional group that can denote any of the following substituents that makes up the entirety of the phospholipid classes. So, for example, if we attach a hydrogen, that's known as phosphatidic acid, and that's important in lipid signaling. We have a choline group. If we attach a choline group, that's important for phosphatidylcholine, which is also important for signaling. And I guess the important point here is that different phospholipids have different functional roles in the cell, and are really, really important in signaling processes that go on within the cell that allow the cell to orchestrate certain processes like apoptosis, mitochondrial signaling, or membrane fusion. So important just to have a sort of a general idea of these structures and how that they can then correlate to the function of the overall phospholipid themselves. So just to go into that a little bit more detail, so phospholipids themselves can be degraded and acted on by a number of phospholipase enzymes, and the most important ones of these to be aware of are A1, A2, and C. And so if we take our structure of the phospholipid on the left here, we can see how each of these enzymes are acting on the phospholipid themselves. And so each of them are going to be acting to essentially mobilize a particular chemical moiety of the phosphate of the phospholipid, and releasing that particular chemical product that can then go on to influence how the cell behaves and responds to signals. And I guess a really important one to be aware of here, I guess they're all important, but one that I've highlighted on the slide here is phospholipase A2, which will hydrolyze this fatty acid here at the C2 position of the phospholipid. And essentially that's really very important in this arachidonic pathway, where we get formation of arachidonic acid from the cleavage of the phospholipid. And that can then, through the action of a series of enzymes, cyclooxygenases and COX-1, can produce various inflammatory mediators that are really, really important for regulating processes like blood vessel vasoconstriction and vasodilation as well. And it's these pathways in particular that are the targets of anti-inflammatories, for instance. So aspirin is a particularly important molecule that targets this particular pathway by targeting the cyclooxygenase, the COX-1 and COX-2 enzyme activities. So just briefly touching on steroid hormones. So steroid hormones are, as I mentioned earlier, are all derived from a common precursor, and that's cholesterol, which you can see here. And in terms of the difference of these structures, they differ in terms of their ring structure, but also the side chains that are attached to it. Okay, so steroid hormones, as I'm sure you're aware, are produced in a variety of bodily tissues and organs, so mainly the adrenal cortex, in ovaries and testes, and also in some peripheral tissues as well, like adipose tissue and brain tissue. And essentially, cholesterol is a really important precursor for the synthesis of a number of biologically important hormones and mediators. So on the left here, we can see that through a variety of different chemical steps, cholesterol can eventually go on to produce the sex hormones testosterone and estrogen. And on the right here, cholesterol can also be manipulated to produce other very important molecules, biologically important molecules, which are the bile acids here that can be listed below. So when we think about cholesterol, mainly from sort of a clinical perspective, it's important to obviously think about medications that can particularly target this pathway, and sort of classically the most well known of that, which I'm sure you're all aware of and have read about, is statins. So essentially, statins are a medication that aims to reduce levels of circulating cholesterol in the system, particularly the low density lipoprotein, which is the cholesterol that we really want to sort of target in the body. And essentially, the way that they do this is that they interfere with the pathway for making cholesterol. So we can see on the right here that cholesterol is actually derived from acetyl coenzyme A, which you may remember from your glucose metabolism. And when you react together acetyl coenzyme A, you form a number of intermediates, and one of those intermediates is the conversion of this structure here, this HMG-CoA, into a molecule called mevalonate. And this is essentially the step in cholesterol synthesis that statins will target. And it's really important that statins target this particular step, because this is essentially the rate limiting step of this reaction. So if we can knock this step out in some way, we're really going to influence the levels of cholesterol synthesis in this pathway. And essentially, by blocking HMG-CoA reductase, which is the enzyme that catalyzes this rate limiting step, we essentially can lower levels of LDL through this mechanism that I've shown here. So essentially, what we have when we treat patients on statins, the statin will obviously act to block this particular enzyme, and that will essentially downregulate cholesterol synthesis in the liver. And this process is essentially controlled by negative feedback. So by blocking cholesterol biosynthesis, we're relieving the negative feedback on the expression of the LDL receptors, which essentially regulates these receptors on the surface of the hepatocytes. And so essentially, if we have more LDL receptors on the surface, we're going to scavenge LDL from the blood more readily, resulting in increased LDL catabolism and turnover, which is essentially the ultimate goal of what we want to achieve, which is to reduce cholesterol levels, but particularly reducing levels of low density lipoprotein and having high levels of high density lipoprotein as well. So just to touch on the mechanism of action of most of the steroid hormones, So these steroid hormones can be thought of as in different categories. So we've got glucocorticoids, which is the classic example of being cortisol. We've got the sex steroids, which are testosterone and estrogen, and then we've got the mineralocorticoids, which is the most classical of those is aldosterone. And essentially, we can think about these functioning in terms of at the cellular level in this way here. So if we think about steroid hormones, so in response to a particular stimuli, they're going to be released from certain tissues, and once they're released from certain tissues, they'll circulate in the blood. And obviously, because they're predominantly quite lipophilic molecules, they need to be transported in the plasma, attached to plasma proteins, so certain globulins that are specific to that particular steroid hormone of interest. So once they're circulated in the plasma, bound to these plasma proteins, the hormones will then be able to freely cross the cell membrane because they're highly lipophilic and so can passively diffuse across that membrane. And once they're inside the target cell, they can then bind to specific steroid hormone receptors that are present either in the nucleus or the cytoplasm. And ultimately both destinations result in the upregulation of gene transcription relevant to the genes that need to be transcribed to carry out that particular biological process. Okay, so that's lipids, and I'm going to very quickly, because I know that we're tight on time, so I'm going to very quickly finish off talking about the carbohydrates. Okay. So carbohydrates, so thinking about sort of simple monomeric carbohydrates first, and we can sort of think about this as sort of the hexose structures. So I'm sure that you're all familiar with glucose, fructose and galactose being the classic sort of carbohydrate monomers. And obviously these are hexoses because they're all 6 carbon structures that are each important components of carbohydrates, and in terms of their structure, very, very rich in carbon hydroxyl bonds, which is where most of the linkage of these monomers are formed to produce the larger polymers. So when we link together these hexose monomers in different forms, we get disaccharides. So essentially we've got lactose here, which is a disaccharide of glucose and galactose, lactose obviously being the sugar present in milk. We've got glucose and fructose, which together, when condensed together, will form sucrose. And essentially very, very similar to amino acids earlier, this is also a condensation reaction where you get elimination of water as a byproduct. Okay, and then just to touch a little bit on different ring structures that you can get with sugars. So there's 2 sort of quite important terms here, which is pyranose and furanose ring structures. So essentially these monomeric sugars exist in an equilibrium, so they can rapidly interconvert between a more open chain, which you can see in the middle here, and they can convert either into a pyranose form, which is essentially a 6-membered cyclical form of the monomer, or you can have the furanose form, which is also a cyclical form but only forms part of a 5-membered ring. And essentially this process of equilibrium between these states enables the formation of 2 different diastereoisomers, particularly for glucose as a monomer. And essentially that's at the C1 position here. So you essentially end up resulting in the formation of alpha glucose and beta glucose. And you can see in the top right hand corner here that alpha and beta glucose are diastereoisomers. So essentially this carbon 1 position here is a chiral carbon, and can flip this OH and H group either vertically or horizontally to form either the alpha or the beta configuration. And this is really, really important because monomers of these alpha and beta glucoses can form the polymers that I'm just about to discuss on the next slide. So when we join glucose units together or sugar units together, we form polysaccharides, so polymers of many monosaccharides. And essentially the alpha glucose and beta glucose monomers that we just spoke about can go on to form higher order structures that are more complex carbohydrates. So essentially these carbohydrates, starch, glycogen and cellulose, are all forms of energy storage. And I guess the key sort of points about these are highlighted in the table here, but essentially we've got starch, which has two different forms in which it exists. So you've got amylose and amylopectin, and amylose is essentially the formation of 1,4 glycosylic bonds, so it forms a relatively linear structure and actually forms more helical structure, so it's very tightly compacted, so it's very good for storing energy, and it's probably the best adapted form of energy storage when we're talking about starch. Amylopectin again is also a polymer of alpha glucose subunits, but compared with the amylose, we have a 1,6 glycosidic bond, so it can also form 1,6 linkages as opposed to 1,4 linkages. And this is really helpful because we can form these branch structures which enable very quick mobilization of energy for release from this structure that can be used to power cellular processes. When we look at glycogen, again, we've also got glycogen as a polymer of alpha glucose, and also glycogen is also a branched polymer of 1,4,1,6 glycosidic bonds. And again, as far as humans are concerned, a particularly important carbohydrate for storage in muscle and liver, and obviously at times where energy is needed, very important that this structure is branched so that the enzymes that are responsible for cleaving these branch subunits can be activated quickly and can quickly mobilize these sugar units for energy utilization in the cell. And obviously cellulose is another polymer, polysaccharide, but instead of being built from alpha glucose, it's built from beta glucose and is more linear. So you get linear 1,4 glycosidic bonds rather than the branch structures that are present in glycogen. So a quick word about glycosylation, so the process of attaching sugar units to particular proteins within cells. And the process of glycosylation initially is occurring on the cytosolic surface of the endoplasmic reticulum inside the cell. So what happens is that particular proteins that reside within the ER lumen are decorated with these sugar residues initially. The enzymes that, once that's reached a certain stage, the enzymes that then further decorate this growing sugar unit are present within the ER lumen themselves. So the growing polysaccharide chain has to internalize and translocate from the cytosol to the ER lumen in order to allow a more complex polysaccharide unit to be formed. Now, when this polysaccharide unit is being synthesized in the ER lumen, there's a number of very specific glycosylase enzymes that are present within the ER lumen and that add sugars at certain sequences of the protein, and that are very specific for recognizing certain sequences of the protein and add sugars in a very specific manner. So there's two different types of glycosylase enzymes that can add sugars to the proteins in the ER. So the first is the sort of the O-linked glycosylases. So these are enzymes which add sugars specifically to serines and threonines within the protein. So this is attaching the sugar units to the OH groups, which are both present in the side chains of serine and threonine residues. And we've also got N-linked glycosylases as well. So these are sugars that are added specifically to asparagine residues that are embedded within specific sequences. So X, N, which is asparagine, X, and then we've got a serine or a threonine at the end. So it has to be able to recognize that specific sequence there. And so just a quick example of the structural role of carbohydrates. So a well-known example is proteoglycans, so glycosylated proteins within the extracellular matrix. So the extracellular matrix, often defined as the sort of the meshwork or the structural scaffold sort of surrounding cells which give it structural stability. And within this particular matrix, the extracellular matrix contains 3 classes of molecules. And you've got your structural proteins here, which are your collagens and your elastins, which help give the structural support to the extracellular matrix. You've got your proteoglycans, which are your complexes which are embedded within the structural network, again to aid support and stability to the cellular cytoskeleton. And then you've got your adhesive proteins for attachment of the cells to the extracellular matrix. So these are your fibronectins and your laminins. And then finally, just to wrap up, arguably probably one of the most important structural roles of carbohydrates is in differentiating between different blood groups. So essentially, we've got 4 different blood groups, AB, AB, and O, and each of them are differentiated by their the attachment of the different sugar residues to the surface of their red blood cells. So, for example, if you have an O blood group, you've got exclusively the O antigen on your surface of your cells. If you've got your A blood group, you've exclusively got your O antigen trisaccharide, in addition to your N-acetyl galactosamine units. And then if you have your B blood group, you've got exclusively your O antigen trisaccharide linked with a terminal galactose residue. And then obviously AB is the best of both worlds. So you have a blood group which is a mix of both A and B antigens on the surface of your red blood cell. And that is and that's it from my end. Thank you all very much for listening. Very happy to take any questions that you guys have, or if anything was unclear at all, do feel free to unmute or pop it in the chat, and we can see what we can do.
Thank you very much, Lee. That was brilliant. I'll just pause the recording.